My name is Martin Smith. I'm the President of Bee Disease Insurance. We're happy to have sponsored this lecture because it's to do with having healthy bees. And bee diseases is about, it's a sort of negative term, but we're keen on healthy bees and a healthy bee population. So we support research into bee diseases and are keen that beekeepers improve their education generally. The healthier bees we have, the better for all of us. A lot of you will have listened to Celia in the past. She's a well-known speaker in this country and abroad, I suspect, as well. Her talks are always entertaining and clear and straightforward. So without any further ado, can I welcome Celia?
Thank you, Martin. Well, thank you, Martin, for that. And good morning, everyone. Nice to see so many of you here. Yes, this is, as Martin says, a talk about bee health. It's called In Self-Defense. Not our self-defense, I hasten to add, but the bees. Now, some of you look old enough to remember when beekeeping was like this. It was a pleasant occupation for sunny summer afternoons with your friends. Very gentle, and as long as you had enough space for your bees and you fed them sufficiently or ensured they got enough food and hopefully didn't swarm, you were assured of a healthy colony and a good honey crop. There were many let-alone beekeepers who never actually went below the queen excluder. And I've met people who say, oh, I don't know what goes on under there. Ooh. And anything to do with disease was usually confined to the back of the book that you bought. And you sort of ran out of steam before you got that far, or, you know, it got a bit boring, so you didn't really bother very much. And really, disease wasn't a big part of our lives. Now, of course, a honeybee colony is an ideal place for the spread of pathogens, disease-causing organisms. It's warm, it's quite humid, and there's all these little bodies crammed together in a small space, all rubbing together and all the rest of it, and exchanging food and doing all these sorts of things. And as we all now know, that is a really good conditions for the spread of diseases. So the fact that most colonies used to remain really healthy and strong is little short of miraculous. Now, of course, times have changed, and now we spend a lot of time, don't we, talking about disease and its monitoring and its treatment. But I am not going to talk about disease today, except a little bit towards the end when I shall have to mention it, but rather how bees maintain their health and what we can do to help that. And as Martin says, that is very important. And it is, I think we should be looking at this in a positive way. You don't all go around thinking, I wonder what disease I'll get next. You do things to try and ensure that you remain healthy. So we'll see how bees do it. And I hasten to add that the more I look at bees and the more I think about them and the more I discuss them, the more alike I come to think bees and people are. And so many of the things that we apply to ourselves, we can apply to bees as well. They're not all that different. So we're going to look first of all at understanding how a colony remains healthy left to its own devices. Then we're going to look at how the beekeeper can help, and a little bit about how the beekeeper can hinder as well, because hinder we frequently do. Now, when the honeybee genome was published, first of all in 2006, and then it was updated later, people were somewhat surprised by the comparatively small number of genes that were associated directly with immunity compared to other insects like Drosophila and things like that, which is the other much studied insect, of course, in entomology. And then it became clear that there is a separate sort of entity as far as bees are concerned, because they are this superorganism. They are this huge family which all lives together. So you've got this big group. So you have actually got some colony level immunity, which is inferred by various things that the bees do as a colony, as well, as we shall see, as an individual level immunity, which applies to bees and is similar to most other insects. So let's look first of all at colony immunity. Now, the first aspect of this, which I want to look at, and I'm not going to go in enormous detail into these, but we'll look at them, each of them in turn, is cleanliness and breeding. Now, bees are clean creatures. They keep their nest clean, they ensure that their larvae are reared in clean cells, and so on. Given half a chance, and as long as we don't disrupt that and cause problems in that relation. So, cleanliness. They actually spend a lot of time cleaning. They use their tongs to swab things down, a bit like mops, and when they use like this anyway, and they use their salivary secretions to do that. And those salivary secretions contain hydrogen peroxide, only a small amount obviously, and hydrogen peroxide is an antiseptic. So that means that the whole of the nest has actually got this sort of antiseptic vibe to it, if you like. And in a natural colony, then, brood is produced always in clean comb, which has been newly produced and is thoroughly clean and disinfected, if you like. And then fast breeding. Breeding, we all quote to people that the queens can lay between 1,500 and 2,000 eggs a day at their peak, which we hopefully are coming up to. And of course, there are also, not as many, but there are a lot of bees dying all the time, older bees, and particularly after the winter, when a lot of the winter bees, or all the winter bees, will die off. And of course, when a bee dies, it either dies outside the colony or it is taken from the colony and its pathogens go with it. So that is quite important in keeping those pathogens down. So breeding is important, and you've got this huge number of new bees coming on, which are hopefully pathogen-free or comparatively pathogen-free. And we do see this with many diseases that bees can recover because of this huge breeding rate in the spring, and of course the fact that many are dying as well. Then there's thermoregulation. Very clever that bees are able to keep their brood nest, particularly at a constant temperature. It's around about 35 degrees Celsius. Not precisely, but that's all right. I'm not good with numbers, so I have to stick with round numbers. So 35 degrees Celsius is a good number to sort of fix. Now, that is too high for certain diseases, and particularly chalk brood, to germinate. Those spores that are often there anyway have to be able to germinate, to grow, to start into growth. And at that temperature, they are not able to start. So they require a lower temperature, about 30 degrees Celsius. Now, it's not a lot, but it is just that bit lower. So that means that a colony which cannot thermoregulate well is more prone to getting that type of disease. Now, of course, that applies to small colonies particularly, nuclei and so on, that find it more difficult to regulate their temperatures because there are far fewer bees to do it. So that's that one. Now, we've talked about the removal of infectious material. The fact that foragers usually die away from the nest. You know, they're a bit like Captain Oates. They go out and they don't come back. So they all die outside. Well, that's fine. But there does appear to be an altruistic element involved as well, in that some bees that are sick will actually voluntarily leave the nest and die outside it. And then there are undertakers who take the dead bodies of any bees that die in the nest out and tip them out through the entrance and frighten the life out of you at the end of winter, because you see all these dead bees. But they are bees that have just died naturally at the end of their lives very often. Not always, but very often. And then sick bees often become foragers earlier. In other words, their development is accelerated, because we all know that worker bees spend the first 2 or 3 weeks in the nest doing all the housework and the housekeeping duties and feeding the larvae and so on. And then at about 3 weeks old, they move outside and become foragers. Now, once they become foragers, they're on the slippery slope to death, because they don't live very long then. They sort of wear themselves out, and their whole metabolism changes, and they don't last very long. So if you accelerate that transition from house bees to foragers, you will shorten the lives of those bees and ensure that they die sooner. Now, there's one good example here, and that's sac brood. Now, you think of sac brood as a brood disease. You see it in brood, and in seal brood, it affects the pre-pupae, that first stage between larvae and pupae after the cell has been sealed. And you think, well, that's a larval disease. How does that affect adult bees? Well, the fact is that adult bees carry it. And it's carried in the hypopharyngeal glands. These are the brood food glands in the bee's head. That's where it overwinters, in those hypopharyngeal glands. Now, of course, if those bees were to produce brood food and feed it to larvae, they will also feed them the sac brood virus if they are infected with it. The bees, the adult bees, don't show any signs of sickness at all. You can't look at a bee and say, well, that's got sac brood, it's carrying sac brood. It doesn't look any different to any others. But what it does do is accelerate the development of those bees, so that many of them never actually feed brood. They transition to becoming foragers. And then they go off out and they're outside, and of course they do die sooner. And they don't normally, when they're foraging, collect pollen. They only collect nectar. And of course pollen is the basis of brood food. So if they were to collect lots of pollen and infect all that pollen, then that would be fed to the brood, so you get the same infection. So that is one illustration. So you've accelerated their development, and you've sort of prevented them from collecting pollen. So that works very well. Obviously, any that do feed brood, or if they actually clean the cells even, then they can transmit the virus, which is why it keeps going. And then there are what I call barrier mechanisms. If you think about a nest of bees, I use the term nest intentionally rather than hive, but a nest of bees. The middle, generally, you have the brood nest. So you have your larvae and your adult bees that are feeding those larvae, and the queen. And then as you move out, you get your bees that actually process the food, and the bees that make wax, new wax. So they're outside the brood nest, and they don't normally sort of interfere with the brood nest. And then, again, as you move gradually outwards into the outside, you get the older bees, the foragers. So there's this gradual movement out from the young ones and the brood outwards to the foragers on the outside. And those groups don't mix to any extent, unless we mix them up, of course. So they don't normally mix. And that keeps the older ones, which are the ones that are more likely to be infected with things, because they bring them in from the environment and so on, away from the younger ones, which have to be protected. And of course, all the larvae are reared in individual little cells. So each of them is in its own little cell, separated from its neighbours, although it's in contact with lots of workers who are in contact with other brood cells. So it's the barrier mechanisms do work to a large extent, I think, and of course everything is a combination of things. And then we have hygienic behaviour. The simplest sort of hygienic behaviour is where bees groom one another. And of course, if they're grooming one another, they can get rid of ectoparasites, these are the parasites outside the bee. Nasty little things that cling onto the outside, like varroa, for example. And of course, some bees are much better at that than others. Truly hygienic bees, what we think of as really hygienic, can actually detect and remove diseased brood, diseased larvae and pupae, even when the pupae are obviously underneath sealed cells. And it's thought that scent is the most important thing here. And in fact, bees that are good at hygienic behaviour have got a better sense of smell than bees who are not. And there's one particular substance called oleic acid, which is a brood pheromone, which is often referred to as the death pheromone. And apparently this is quite an important pheromone, which tells the worker bees this larva, pupa, is in trouble and is sick or dead or whatever. And then there are the use of antimicrobial substances. Now, we've already mentioned hydrogen peroxide. as being present in the hive environment as a whole. And then there are substances added to the bee bread, the stored pollen and so on, from the bee's gut. Now, the bee's gut does produce, or the bacteria in it, produce some natural antibiotics. So those are incorporated into the brood food. And these can have obviously a good effect on the brood. And then the last thing, which is really part of this, is propolis. Now, I expect you all hate propolis, but, you know, we all do, don't we? It gets everywhere, doesn't it? But it's extremely important in the bee community. It's antimicrobial, and of course the bees actually steal it because the plants produce it for their own use to protect themselves from attack by microbes of various sorts, and the bees steal it and use it for themselves. It isn't of constant composition, it varies because it varies depending upon the plants it's come from, but it is antimicrobial. And if you look at a natural bee's nest, a natural habitat for a honeybee colony is perhaps in a hollow tree, and you will find that the whole of the inside of that cavity will be covered with propolis. Now, partly, of course, that seals it so that you don't get splinters and bits of wood breaking off and things like that. But it also forms this sort of envelope around the nest, this antiseptic envelope. And they also deposit quite a lot of it around the entrance, where the bees sort of come in and out all the time. So it's very, very important. In fact, there's some work going on now experimenting with having rough insides to hives to encourage the bees to cover the sides of those hives with propolis, or screens that you can sort of fix onto the sides of the hives. Various ways people are trying to do it. But the theory is that you know, if you surround them with propolis, then they're likely to be healthier. So one can question whether we should be scraping all this propolis off and cleaning all these insides of hives. I don't know. Depends who you listen to be honest, whether it's a bee inspector or a scientist. I don't know. But anyway, so propolis is lovely stuff. There it is. Bees collect it in their pollen baskets, of course, but they can't get rid of it. They have to have it chewed off, so they have to wait patiently while other bees chew it off when they get back to the colony, because it's such horrible sticky stuff. And that's how we usually see it on a warm day, when it gets all over our hands and the steering wheel and the gear lever and, you know, the handles at home, and our nearest and dearest gets a little bit annoyed because everything gets sticky and brown. So anyway, that's propolis. So those are the ways in which the colony as a whole is protecting itself. So lots and lots of different techniques there. So if we go on to individual immunity. Now, this is really interesting, and I'm going to look at it under a number of sort of headings. I'm going to look at the cuticle first, then gut structure and the environment, because the gut is the commonest way, or one of the commonest in which pathogens go in, obviously, in the food. And then the hemolymph, and finally we'll look at the production of antimicrobial peptides, which are quite interesting things. No, it isn't finally. Finally is vertical transmission of immunity. So that instead of being transferred from bee to bee, it is bee to bee, but it's sort of downward, so it can be from drone to queen, queen to eggs, and so on. So it can be in different ways. And of course, microorganisms can be transferred in the same way. So we're going to look at those in turn. Now, the cuticle, first of all. The cuticle forms an impervious layer over the bee, and its main job, it's like your skin. Its main job is to keep things that should be outside, and keep things that inside should be inside. And one of its main jobs, of course, is keeping water in, because this is a land-living animal, and water loss is very important and can cause a lot of damage. So it's very important from that point of view. But it's not, of course, completely solid. If they were covered with this rigid cuticle all over, then they wouldn't be able to move, they wouldn't be able to breathe. So it's got these little membranes between the plates of cuticle. So they're all in plates. Get the right one. So you can see that these, they're not, you can't actually see, they're not plates really, you can't see them, but they are there. Each of these segments has got a plate at the back and a plate underneath going down the abdomen. Thorax is far more complicated. But the abdominal plates have all got little membranes between them, little membrane bits. They're only narrow, but that enables the plates to move relative to one another, not that much, but relative to one another. So that the bee can breathe and can move and all the rest of it. And of course they provide a way in for things, some things like varroas, for example, that can actually pierce those membranes, whereas it wouldn't be able to pierce the hard exoskeleton cuticle of the bee. But as long as the cuticle remains undamaged, then it is a very good protection, like having a suit of armour, really. But of course, it can have broken hairs, for example, and of course, if something like varroa pierces it, then that can be a way for other things to go in. Then there's the gut. Now, you haven't had your lunch yet, so you're all right. This is a dissection. It's been pulled out of the bee, because if you just leave it in the bee, you can't really see anything. And we can see, I expect you're all familiar with this anyway. You're all knowledgeable, aren't you? You look a knowledgeable lot. The first bit of it is the crop. Now, the crop there is collapsed. The crop can expand and collapse. And that is the part that the nectar or water that the bees is getting from the flowers or next door swimming pool or whatever it might be. That's the part it goes into. And of course, when it's collecting nectar or water, that crop expands so that it can carry it back to the colony. Because inside the crop, just jutting into it, and I don't know how well you can see that, is this structure here. You see that white sort of ball? You can just about see it, I think. Should have put a circle around it, but I haven't. That's the proventriculus. And that is a one-way valve. It's a one-way valve. It's got flaps with little pockets in and bristles and things. It's a very, very crafty bit of engineering, really. And it's able to close the crop off so that the contents of the crop can remain in the crop. But it can release things through. And it can trap the pollen grains as well. And it releases those through because they need to go through into the next part of the digestive system, which is the ventriculus. That's the big bit that goes all the way around there, from there right through to there to the small intestine, which is here. Now, as well as some pollen, obviously the pollen is trapped there, but also that proventriculus is able to trap some spores, disease-causing spores. And that is also quite important because when the disease-causing spores are passed through into the ventriculus isn't a very friendly place. Slightly acid and all those digestive enzymes, because that's where all the digestion goes on. The ventriculus is the same as your stomach, or similar anyway. And that's where all the digestion goes on. So, you know, it's not a good place to be if you're a little spore or something. So that's that one. And then if we go on, oh, there's the food, yes, I'll come back to that and why that looks like a sort of lump of food. It's mainly pollen, of course, which is white yellow, but that's the food down there, and you'll see in a minute why it's in that sort of form and not just spread out around everywhere. So if we look at a section, so we're going to cut across the ventriculus and then look into it, and we get a section. And this is the most exciting slide you'll ever see. There. Section through the ventriculus. Sorry about that. Now, of course, the digestive system is surrounded, first of all, with muscle. I haven't put those in. But it's got these muscles around so that it can squeeze the food along. Your digestive systems are just the same. The food squeeze called peristalsis, if you can remember back to your biology lessons. The food is squeezed along, otherwise it would just sit there, wouldn't it? It wouldn't go anywhere. So you've got that layer of muscle. And then inside that, you've got this single layer of cells, the epithelium, the gut epithelium. So the gut epithelium gives you a bit of a barrier for things. Remember, the gut is completely surrounded by hemolymph, the bee's version of blood, but of course it isn't red. It's completely surrounded by hemolymph. It's all bathed in hemolymph, so it's not sort of just sitting there in isolation, as it were. So those cells in the epithelium are very important because as well as providing a barrier, it's the place where the enzymes are produced in that epithelium. It's also, in fact, I have to mention disease here, but it's also the place that nosema attacks. So nosema attacks that, which obviously affects the digestive process because it affects the production of those enzymes. And it also damages it and it allows easier passage of viruses, for example, because it damages that epithelium. So that's quite an important layer. Now, inside that is this structure, which is called the peritrophic membrane. Now, it's not really a membrane as we think of a membrane. It's sort of gelatinous. And it's produced along the whole of the length of the ventriculus, continuously, which is why it looks like a series of sort of more or less concentric circles, because it goes on being produced, so the next one's produced and wraps around the previous one and so on. And again, they're pushed along with the food in the middle, which is why the food look like that. We call it a bolus of food, a sort of lump of food, if you like, which is in the middle of that peritrophic membrane. Now, the peritrophic membrane, it probably protects, the general view has always been that it protects the lining, the epithelium in the gut from any sort of damage from spiny pollen grains. You know, lots of pollen grains have spines and they're quite hard and so on, so it can protect them. But of course, it also forms another barrier to things. And the interesting thing is that it takes a little while to develop in both the larva and the adult, because larvae have them as well. And in the larva particularly, in the very, very early stages of larval growth, before the peritrophic membrane has developed, it takes very, very few spores of AFB to infect a larva. Whereas later on, once the peritrophic membrane has developed, then it takes an awful lot more of those spores to cause any infection. So, you know, it does protect from a lot of the microorganisms. The only problem is that it isn't completely impervious, and neither is the epithelium. They can't be, because they have to allow the passage in of the enzymes which are produced in the epithelium. So that has to come through the peritrophic membrane, and then the digested food, the simple molecules, the things like glucose and amino acids and so on, have to be able to pass out into the hemolymph so that they can be transported to wherever they need, all the cells in the body. So it is not completely impervious to things going through it, but it does form a major barrier to a lot of things. So that's the inside of the gut. And the other thing we have to consider is that there are bacteria and fungi living in the gut. Now, most of them are actually in the hind gut, which is the next bit after the ventriculus. But there are lots of them. They're the same, many of them are the same ones as you've got in your guts. Exactly the same species. And they help with digestion, and they actually produce some antibiotics, natural antibiotics of their own, and some vitamins. So they are quite important. So those antibiotics, again, can help to combat anything nasty that comes in and gets past the earlier parts of the gut. And then we go on to the hemolymph. So the hemolymph, the bee's blood, if you like, which does all the same sorts of things as our blood, except that it doesn't have a respiratory function. That's why it doesn't have any red hemoglobin. Our blood carries oxygen and carbon dioxide around and is a very important part of the respiratory system. In the bee, the respiratory system is slightly different. So the hemolymph does everything else, but it doesn't actually take part in that. So it's a clear fluid, as you'll all know. I often think it's a shame it's not bright red, because I think we'd take much more care of squashing bees if they spurted bright red blood at us. But that is simply I have these strange ideas, but there you go. So hemolymph, very, very important in the immune response of individual bees. It contains cells called hemocytes. Now, hemocytes are a real problem because there are different sorts, and different authors give them different names. Sometimes the same name for different ones with different functions, or sometimes different names for the same one. So it gets quite complicated. In fact, there's a whole paper, I can't remember who it was by now, written about hemocytes and their sort of nomenclature. very boring reading, but you know, it's one of these things, quite complicated they are. And the cells are obviously very important, and their highest count is in larvae and pupae and very young bees. Then they start to decline. In other words, the immune responses of older bees are not as good as young bees and larvae and so on, which is probably what you'd expect. But they go on being high numbers in the queen. She's well protected. Now, what we have to think about is how this all works. Now, when an invader, which we'll call a pathogen, an invader, comes in, gets into the hemolymph, it actually has on it these pathogen-related molecular patterns. Now, that's quite a mouthful. Now, this is where COVID is really, really useful, because you've all been used to seeing pictures of this little virus with all these spikes. And we all know how important those spikes are. Well, it's the same sort of thing. It's a pattern on the outside of this microorganism, whatever it might be. And those bind to what we call pattern recognition receptors. That's just a long name, and you don't really need to know the names, but it binds to these protein substances actually in the hemolymph. So it's bound to that, so they've recognised, the hemolymph has recognised that these are invaders. They shouldn't be there. They're foreign bodies, if you like. And once that's happened, it triggers a number of different reactions. Now, I'll just summarise these and then I'll go through them. Phagocytosis, first of all, and that one it will be easy to understand. Then the phenol oxidase cascade, which is probably slightly more of a mouthful, and then the production of antibacterial peptides. So let's have a look. That should be antimicrobial, actually, not just bacterial, because they attack other things other than bacteria. So I don't know why I put bacterial there, but it should be microbial. Anyway, let's look at those one at a time then. Now, phagocytosis, this goes on in your blood, exactly the same. There are hemocytes, blood cells. In this case, they're called granulocytes, but they're sometimes also called phagocytes for obvious reasons. Doesn't matter. But those cells surround and kill invaders. So you can imagine they rush to an invader and they surround it and kill it. So that's your little disease-causing organism. And that process is called phagocytosis. Very easy to understand, very straightforward. It's exactly the same in your bodies. So, you know, that's what goes on. But then the next one, phenol oxidase. Now, phenol oxidase it's actually an enzyme, and it's produced in other hemocytes, and it regulates melanin production. Well, melanin is the dark pigment that is laid down in the cuticles, what makes the bee dark-coloured, the melanin, and phenol oxidase is an essential part of that. Now, the phenol oxidase is not floating around in the hemolymph in that form. It floats around in the form of prophenol oxidase. So, in other words, it has to be converted to the active phenol oxidase before it's functional. And this actually attacks the nodules which have been formed in phagocytosis, and it lays melanin in them so that it encapsulates them in a capsule, so that they're sort of dark and trapped, and that's it, that's fixed, it's not going to go anywhere soon. So that's what the phenol oxidase does, a very important process. And in a bee which has been infected multiple times, you can detect these as little black blobs in the hemolymph. Now, of course, all these things we have to remember take energy, and they need protein as well, usually. Well, this does, because it's a protein substance, an enzyme. So, you know, these are all sort of taking things from the bee which it could use for other things. So any immune response is going to cost the bee in terms of energy and substances, basically, protein. And then the final one is the antimicrobial peptides. Got the right name here, you see. Peptides are parts of protein molecules. They're not whole protein molecules, they're smaller. If you chop the protein molecule up, you will get peptides. Now, these are antimicrobial peptides. Now, they are protein substances, as I've just said, but they're manufactured in the fat body. Now, the fat body, most people don't pay a lot of attention to the fat body because it isn't really a body, it isn't really a structure. It's sort of spread out, it lines the inside of the bee. And of course, sometimes there's a lot of it in some stages, young bees, larvae. Sometimes there's a lot less in foragers and so on. And it does all sorts of jobs. It has been compared to our livers. So it can detoxify things, it produces things, it breaks things down. It's a really complex tissue. And I think it's a pity it's called the fat body because it sort of suggests a lump of something, and there isn't a lump of something. But anyway, this is where these antimicrobial peptides are made in response to this trigger from the patterns on the invaders recognised by the proteins in the hemolymph. And they are persistent. Once they've been produced, they stay in the bee. But they are only produced in response to infection. So they're a bit like our antibodies, but not the same. They're very similar in the way that they work. One of the things is that they're not specific. You know, our antibodies are all specific. If you get an antibody to protect you from flu, then it won't protect you from COVID, or it won't protect you from, you know, any of your dozens of things you can think of. But these are not specific, so they can protect from other infections other than the one that has actually triggered their production. So they're very, very important. And it says there they can be passed from bee to bee. So they can be passed in brood food, for example, from bee to larvae. So that, again, it's quite a sophisticated way of doing things. And trophallaxis. Bees exchange food all the time with one another, so that they can transmit these things because they're in the gut, they're in all the secretions from the gut and so on. And then there's vertical transmission of immunity. Now, this is something that's a fairly new idea, most of this. It can be transmitted, we've seen, from worker, nurse bee to larvae. That's a very simple way of seeing it. But you can also transmit it from queen to brood. And there's our pathogens, and we take this as the queen. And those pathogens can get into the queen either by contact or in royal jelly. Now, of course, if they go in royal jelly, then they go into the gut, which is fine. And they can get into the gut anyway, even if they go in by contact. And they go into the queen's gut where they are broken down. Now, they're broken down into fragments of pathogens, which in themselves are probably perfectly harmless and aren't going to do anything. But those pathogens go into the hemolymph, and then they bind to this substance called vitellogenin. Vitellogenin is a really, really important substance in the honeybee. It's found in all egg-producing animals, in fact. Well, I think that's correct. It's virtually all anyway, if it's not quite all. And it's actually used as a precursor for yolk in eggs. And that's its prime function. But in the bee, in the honeybee, because most of the worker bees don't produce eggs, so it's been sort of hijacked to have other functions as well. But in the queen, it is still used as a producer of yolk. So having bound to this vitellogenin, it's a very complex substance actually. It hasn't got a constant composition. It's what's described as a glucolipoprotein. That gives you a good idea. And the protein is probably the most important part of that. It's a very important protein substance. Very important, for example, in winter bees as a food store and so on. So it's a very important. If you don't know anything about vitellogenin, well, find something that describes it and learn, because it is really fascinating and worth knowing a bit about. So these fragments bind to those vitellogenin molecules, and then they go into the yolk, which are put into the oocyte, which are the earlier stages of the eggs in the ovaries. Those become eggs, and then, of course, those develop into offspring, into larvae. And because these fragments of these pathogens are there in a sort of inactive state, if you like, the larvae are able to initiate these immune responses. So they're able to generate the antimicrobial peptides and so on that will protect them. So it is a bit like vaccination almost in that sense. And it's a very, very important way. Now, when this was first described, it was Salmonella, Amdam and Freitag. In 2015, that was the first sort of paper that was produced on this, and there were a lot of people who were very sceptical about it. And I remember asking one chap, and I can't remember who he was, about it. Actually, it's a spring convention. Oh, no, it's all untrue. That doesn't work, doesn't happen. But I think now we know that it does happen, and that it is probably quite important method. Okay, so we've got all that. We can see that that is very important. And we've got the various ways that the colony and the individual protect themselves from any sort of infection. So let's have a look at beekeepers. Beekeeper, of course, is the bee's biggest enemy. You know, left to their own devices, they got on perfectly well until we came along. So beekeeper effects. Now, I shall sort of go through this a little bit quite quickly. These, you can link these back, if you can remember that far, to the things I said at the beginning. So, rough handling, damage the cuticle, upset the bees, loss of heat every time we open a colony, unless it's really, really hot, which it isn't all that often, is it? Then they lose heat, so that's a problem. They've got to restore all that. We unbalance colonies, we make up nukes. We make up mating nukes, would you believe? That's an unbalanced colony, if you like. We do various things. We move frames in and out and so on. So unbalancing them so that there are not the right proportion of brood to house bees, nurse bees, and foragers. You need that right mix of bees in order for the colony to survive well and look after itself. And we compromise some barriers. Now, as soon as the weather really warms up, I'm hoping next week, I'm intending to shook swarm my colonies. I've never actually shook swarmed my colonies before, but I did a lot last year because I helped with the Stoney outbreak, which many of you will have heard of. So I know how to do shook swarms now. But I intend to do that. But that is really unbalancing a colony and compromising the barriers as well. When you shake all those bees together, young bees, old bees, the whole lot, in they all go. At least there's no brood. But then, you know, you really mix them all up. So anyway, compromising some barriers. And we do that all the time. Even when you shake a frame off, you know, if you take an outside frame off, which is mainly going to have older bees on it, and shake it over into the brood nest, which should have young bees in it, you are immediately mixing old bees up with new bees, young bees. So that's another way of doing it. Remove propolis. We've already talked about propolis, and we scrape it off, we throw it away, we burn it off, we do all sorts of things with propolis. And maybe it would be better left, but I'll leave you with that thought, I don't know. And then we introduce pathogens. I think we've all learned that we need clean gloves, disposable gloves, but there are some people who still don't use them. And I'm particularly horrified by sometimes you see people on the telly who've taken up beekeeping. You know, we've all seen them, haven't we? These celebrities or gardeners or whatever, and they're going to have a hive in the garden. And they're usually equipped with nice big leather gauntlets. Very, very bad things. We know that's important. But our clothing as well, and anything we use in the colonies, like bee brushes. Bee brushes, you brush these bees off, and you go to that hive and you brush those bees off. You know, all these kinds of things, hive tools, the whole caboodle really. Anything, if you've got disease organisms, can spread and cause those disease organisms to be spread to something else. So we have to think. An outbreak of EFB does actually concentrate the mind wonderfully and make you realise, you know, well, we could do this and we could do that, and we're not really careful enough about this, that and the other. So, yes, introduction of pathogens, quite easy and comparatively easy to actually stop. And we cause stress. We cause enormous stress on the poor little things. And I'll come to those in a minute. And finally, use of antibiotics. Now, we're lucky in this country, we don't use antibiotics. I don't think we use them anymore at all, do we now? I don't think they're used even for EFB, even in some cases. They were a little while ago. Oxytetracycline used to be used for EFB, but it isn't. Now we use shook swarms and destruction, things like that. That is much, much better than antibiotics. Antibiotics are terrible things. I was over in the States a couple of years ago, and well, it's 3 years ago now, and they just announced that all the beekeepers over there, if they wanted to use antibiotics, they had to get a prescription. And they were in a terrible state. The commercial boys were stockpiling antibiotics like mad before the law changed, because they are so used to using antibiotics for AFB. And of course, it doesn't kill the spores. So the spores are still there inside the colonies. It only kills the vegetative part. It keeps it suppressed. But as soon as you stop using the antibiotics, it just flares up. So that is a real problem. But if we look at antibiotics and we look at some of the other problems, because there are lots of beneficial bacteria in a beehive. And of course, what do antibiotics do? They kill bacteria. So they'll kill beneficials as well as damaging ones. There are ones in the gut, we've already talked about those, and used in the manufacture of bee bread. We've already mentioned these. These are the things they do. And you've got this wonderful lactic acid fermentation which goes on, which preserves the pollen that comes in and changes it into this bee bread, so-called, which is a bad name, but still it's there and it's stuck. So beneficial bacteria are important, and we really don't want to go down the road of using antibiotics. Well, we won't now, I mean, obviously. I remember years ago, it was at Stoneleigh in the Dixon Pavilion, for those of you who can remember that far back, so it's a long way back. And Mark Winston from Canada, he wrote a very, very good book, which is getting a bit out of date now probably, on bee biology. And I remember him sitting with a group of us there and saying, I do wish we'd gone down the same route as you did with AFB. He's from Canada. You know, he said, because we've got so many problems. And he said, and you've managed to keep it to a very low level with the destruction policy. Yes, the destruction policy is hard for the beekeeper. I'm not saying it's nice, but it's not. And having done it and all the rest of it, it is horrible. But it's much for the greater good. And it really is a good way of controlling these things rather than recourse to antibiotics. Okay, so how are we going to help them? Well, there are three ways really here that we need to think about. Ensure good nutrition. Here's the mention of disease, I'm afraid. You can't go through a whole lecture without talking about varroa at some point, can you? Varroa and nosema, you've got to control those and keep stress to the minimum. So if you do all that, now I mean, apart from the varroa and the nosema, this is what we think about with our health, isn't it? We all try and eat well, and we try and keep stress to the minimum, because we know that stress makes us more likely to get disease, to get ill, and bees are just the same. So let's look at those in order. Now, of course, they need nectar, which is their staple energy food, but nectar does only supply energy. It doesn't supply much else. I know there's a few other bits and pieces in there, but they're not really significant. Pollen is the driver for colony health and colony size and everything else, because it provides the protein, but it also provides the fats, the vitamins and the minerals. In other words, if you want a balanced diet, particularly for your young, and remember larvae are young animals, and like any young animal, they require a balanced diet with all these nutrients in it, then you need pollen. And pollen is absolutely vital. Bees need, from their pollen, they need a percentage of about 20% crude protein. That's just a measure of how the protein content. And they need a balance of essential amino acids. And essential amino acids are these, they're building blocks of proteins, amino acids. They're the blocks that go together, and there's only 22 of them, but you can combine them in any order, any number, and they make up all the proteins that we know. But there are 10 of them which are essential. They have to be taken in that form, the form in which they are complete, if you like. They can't be changed at all. The same 10 that you need, most other animals actually need the same 10. And they have to be balanced. They have to be the right amount. So no, we're having loads of one and not very much of another. So that is important. And we now know that bees benefit from a mix of pollens. So if they're stuck on a monoculture for a long period of time, that is not good. They need a mix of pollens from all sorts of different sources. And if you look at some pollens, because pollens are not equal, you might see pollen going in and think, oh, that's brilliant. but not all pollens are equal. And this just shows crude protein, doesn't show anything else. But you can see something like Viper's bugloss, brilliant plant Viper's bugloss, all the echiums are, and there is some echium, well, there were some a year or two ago, being grown commercially for oil. Wonderful things. It's the same family as borage, things like that, which we all know are good. Apple's good, white clover's good, oilseed rape's good. Those are all above 20% crude protein. And then you come on to the others. Now, obviously, maize and pine are wind pollinated, so they have not got very much protein in them, really, although the bees will still feed on them. And I've watched bees feed on maize pollen, collect maize pollen avidly. You know, if there's nothing else much about, they will collect it. The dandelion is not very brilliant, but in fact, dandelion is very attractive to bees because the outside layer of the pollen grain, the pollen kit, has got a lot of fat in it, lipase, and that attracts the bees, apparently. They like that, so they go for that. Obviously, it's got quite a bit of pollen in it. It's also deficient in a couple of amino acids. So dandelion pollen on its own is not brilliant. So what you need is a mix. And there's some work being done on the benefits of polyfloral pollen. It causes an increase in glucose oxidase, which is the enzyme which produces hydrogen peroxide. So that's important. It increases the hypopharyngeal gland protein, so the protein in the gland that's going to produce the brood food to feed the bees, the larvae. Increase in phenol oxidase activity. Remember, this is the enzyme that actually encapsulates those microorganisms and renders them inactive. Increased colony growth, obviously from the increase in the hypopharyngeal gland, and the basic immunocompetence is increased. So the bee's ability to withstand infections is increased. And this was some work done in 2010. So yes, that's very, very important. Should have a mix of pollens. Varroa, sorry, but Varroa, it is the only slide on Varroa. Varroa is the key to a lot of things. Feeds on the proteins in the fat body, lowers the bee's immune response, and it spreads viruses, and we know that's not going to be good. So, you know, we need to keep Varroa low. We need to control nosema. It's a slide of nosema. There's a few spores on there. There you go. Those are the spores, those little white things up there. You need a microscope to see them, obviously, because, again, it damages the gut, uses protein and energy, causes premature aging and death. Every disease that bees get kills them sooner than they would die otherwise. Maybe quickly, maybe slowly, but it kills them. And it reduces immunity again, particularly Nosema cerani, reduces the immunity of the bee. And of course, they all actually transmit viruses and introduce viruses. And we've got all these from Varroa. Deformed wing virus we're all familiar with other people's bees, of course, we've seen it. And in Nosema apis, black queen cell virus, B virus Y, and filamentous virus. Black queen cell virus, a particular problem to major queen producers. And then finally, stress factors. Now, I won't hesitate over this, I shall go on over my time and they'll shoot me or something. I don't quite know what happens if you go over your time. You shoot me, okay. So I've got to be quick. Now, there are environmental ones, weather and so on. You can't do much about that. Nutritional we've talked about, mainly pollen. Beekeeping operations. Everything you do stresses your bees. So, you know, it's up to you to learn to handle bees really, really well and know when to look at them, when to open hives and when not to. Migratory beekeeping is terrible. And obviously most of us don't do it, but in this country they don't actually go very far anyway, even commercial beekeepers. Terrible thing. I won't talk about America. Gets me going. And I haven't got time, fortunately. And chilling colonies, we've also talked about, and creating unbalanced colonies. So all those cause stress. In-hive and agri-chemicals. Beekeepers like to shout a lot about agri-chemicals, but remember that most beekeepers put chemicals in their hives, and the commonest chemicals in beehives are in-hive chemicals. All right, agri-chemicals, we could talk about those till the cow comes home, but, you know, hive chemicals are also very important and will have a stress effect. And we all know, you know, even Apiguard, stop queens laying and so on. So, conclusions then, just very, very briefly. Bees do show immunity to disease. They show it at the colony level, lots of different ways, and at the individual level in quite a few different ways. And we can help by ensuring they've got good nutrition, controlling varroa and nosema, because those are, I'm afraid, we've got to think about those as diseases because, of course, varroa is always with us. And nosema is incredibly common for most people. And reducing stress. And if we do all that, and we think about it all hard, try and think, you know, they've always been subject to stress, then we will have healthy bees, happy bees, we think they're happy, who will get bumper crops of honey for us, and will be free of disease, we hope, will not need BDI. No, I shouldn't say that because they have sponsored me lecture. So, you know, there you go. That's me finished, I think. I hope you've enjoyed it and perhaps have learned one or two things. Thank you.
Well, if we're that successful, we can eliminate disease and eliminate BDI, then I think we would regard that as a success. We're supposed to finish at past.
Yes, okay.
So we've got precisely half a minute of questions, but we'll run on for 5 minutes.
Hello. Forgive me, I am quite new to this, but I wonder, when you were talking about in the bee gut, about surrounding the pathogens with the black stuffs and then like transmitting it between them, is there a kind of case there for perhaps isolating a bee or two and subjecting them to those things and then putting them back in the hive as to the, like, inoculate the rest of the bees against a lot of viruses all in one go?
Can you answer that then?
Well, it'd be a nice idea, wouldn't it? You just imagine, yeah, it's the same idea as sort of inoculating us, vaccinating us. You know, you're building up the antibodies, in this case the antimicrobial peptides and so on, which you can then take out and put in another individual. But the thought of actually, how would you actually pass it on to the other bees? He doesn't know that, he's new, he says to this. Practically, I mean, theoretically, yes, but practically, someone else cleverer than me would have to think that through, I'm afraid.
Okay, thank you.
Thank you.
Thanks, Celia. I just wanted to ask about your views when you were talking about the pollen needs. What are your views on pollen substitutes?
I've used, I mean, there's pollen supplements, which include some pollen, for those people who aren't familiar with this, or there are pollen substitutes, which are made completely from other things like soy flour and baker's yeast and things like that. I don't really have an answer to this. I've only ever used them once because everybody keeps asking me this question. So I thought, well, I'll buy some and try them. And I mean, it wasn't a controlled experiment, but I tried it on a couple of hives and left the rest. And I couldn't see any difference at all. I mean, theoretically, yes. You could use these in the spring to build up a colony if you wanted an early colony, like now when the oilseed rape's out and you want it really built up in time. Or theoretically, you could also use them in the autumn or late summer, so you could build up the supplies, the stores of vitellogenin, the protein stores, in the winter bees, which would help your winter bees. So there are two periods in the year when you could theoretically use them. Hopefully, you know, you've got your bees in an area where there is sufficient supply of pollen. That can be a problem because these days, and particularly we're seeing it in London, in the centre of London, where you've got this huge number of bees, which are on roofs and all over the place, and there is not sufficient food to go round. There's not sufficient forage because pollen is finite. Once a flower produces its pollen, then it ain't going to produce any more. Nectar it can produce some more usually, but pollen it can't. So you have actually got a finite amount. And in London, they've seen the yields of honey going down because the colonies are not growing so well, and they've also got huge problems with the FB, I believe.
Couldn't possibly say.
No, all right, you can't possibly say, but they have, I know, because I've got friends down there who've told me they have. So, yeah, so, you know, this can be a problem. So as long as you've got your bees in an area where there is adequate supply of different pollens, then that is fine. But if you want to, I mean, in the States, they use pollen substitutes absolutely automatically to build up their colonies, because they take nearly all of them down to almonds in February.
Is that a reason why you don't recommend it?
No. I have nothing against Americans, nothing at all. I've got some friends in America, would you believe? But no, some of their beekeeping leaves a bit to be, not my friends, but we won't go into that. But yes, you know, where they've got to get their bees ready for a crop at a certain time of year, early in the year, before bees would normally have been built up in most parts. I mean, obviously the country varies, but some parts of the country. You think it's something like, I think it's about 80% of all bees go down to the almonds in, it might be higher than that actually, in February for pollination. I mean, you know, they're going to be coming from places which are pretty cold and bees are not going to be built up in time. So they use it automatically over there in large quantities. And there's always a lot of discussion goes on about which are the best ones. But yeah, so I don't know, you pay your money, takes your choice. If you think they help, well, use them, you know, try them. I remember one day saying, before I tried them, sorry, he's going to stop me,
We're not going to get time to go all the way around there anyway, so I'm afraid it's going to be the last question. If you've got a burning question on that side, then perhaps Celia might stay for a few minutes.
That might be.
There's a chap there who's got the mic already, sorry.
Right, Celia, shook swarms. Don't be frightened. The lady who introduced me to them, Jill Brewer, many years ago, which I think you already know, I was very frightened to do it, but they work so successfully.
Yeah.
And of course, you keep your hives so clean. You have basically, you're starting from A1 with a full colony.
Yeah.
And I've so far found them very, very successful. Just got to be brave to get rid of the nice clean brood you see.
I have been involved with them because I helped with all the ones at Stoneleigh. There were 16 colonies. We shook swarms at Stoneleigh when they had EFB last year.
And I'll give a small plug for
very
BDI as well now. Obviously, we are doing our shook swarm. If you are unfortunate enough to get EFB, then in prior years you would have only had the frames and the bees of that specific colony destroyed, whereas in this case, the frames. In this specific case, if you have EFB in your apiary, unfortunately enough, then we will, in conjunction with the inspectors, compensate you for the equipment that is destroyed across everything that's in the apiary.
So the whole apiary is shook swarmed, apart from the disease one which was destroyed, but the whole apiary was shook swarmed. But yes, you get compensation for all those. This is a sort of, is it still a trial?
So it's a two-year trial.
A two-year trial.
This is the beginning of the second year.
Yeah.
And if your colony is it's later on in the season, it's August or whatever, it's too late, then the shook swarm would happen the following spring.
Yeah. Well,
anyway, that's enough of a plug from me.
Okay.
I'm sorry if you didn't get a question over that side, but can we thank Celia again? Yes, and she's here. Thank you very much.